भाई मतलब किसका है? नया है। हाँ नया मतलब उसे दिया था मैंने पहले यूज नहीं किया। यूज नहीं। हाँ। क्या? अरे थैंक यू होगा। मैं नहीं खाना उसे फ्लावर में होना चाहिए। छुट्टी है खाने ही वाला है। भाई सच्ची में वाला बहुत कम क्या तुम रख रहा है? तभी ले रहे हो उसको। नहीं आया। मेरा भी नहीं है। आज मेरे पास एग्जैक्ट प्रेडिक्टिव बस इतने के खा लेना एक भी कम फिर चिंता में जाना है यार मुझे तो क्या मूवी है क्या मूवी सही है ना मैं मूवी के लिए बोल रहा हूं तू कैसा नहीं देना पड़ेगा एकदम मुझे पेट भर जाएगा। अरे पेट खाने के लिए कौन खाता है तो ऐसे ही खाते हैं वो तो वाले बनते हैं तांबा देखी तेरी ऐसे करेंगे मेरे से अलग है उसमें सही अलग अलग चलो हर दिन अलग अलग रेडी कर देता हूँ एक ए बड़ा फिर एक एच बड़ा नहीं आता हो कभी नहीं आता हो हर दो बार सो जाऊंगा हिंदी कंप्लीट हुआ रोज अपना किस वाला मैंने भेजा लड़की ने लोग के लोग के और बात करेंगे और बात करेंगे तो पूछे आ रहा है मुझे किसका खाओ विलेज का दोनों भी सेम ही से आ रहे एक ही बोल यार एक ही बोल मारा हाँ दो भी दो बातें भाई मतलब और क्या रहा था उन्होंने दो बार जीते हो एक मैच हमें टाइम नहीं मिला है क्या ही करती या चीज खाएगा नहीं या चीज खाएगा नहीं अभी कुछ अलग खा लूंगा टाइम आ जाता है मुझे ऐसे नहीं है श्योर है मैं तो वो क्या कर रहा हूँ पता है दूसरा हफ्ता है उस मेरी लड़की हो गया मैंने सुबह से किया है दो नहीं अरे जरूरत तक आए नहीं नहीं रहा वाली अभी तो तू आया है भाग जा इधर तो इधर भाग गया है तो रिक्शा से भाग जा मैं रेट निकला है जल्दी निकल जाना है मेरे गाड़ी नहीं रमजान हो गया ऐसे मेरी वाली 4 मिनट आ गए
ये पूछा
नहीं मैं बता रहा हूँ मेरे सामने तो नाम जाती है
पूछा
नहीं तो बता देखो बता
क्या हो गया बता मेरे बेटे बहुत मेरी कौन नहीं बोल तू नहीं करेगा
उतना अच्छा नहीं है मेरा वो एक्सेंट
तो एक्सेंट उसी का बेस्ट है तू बोल के दिखा लेकिन मैं फिर आगे नहीं
नहीं अरे ना वो एक्सेंट नहीं है अच्छा
हां लड़कियां बोलते हैं तो आराम पर ही देते मुझे एक्सेंट
अरे बहुत मुझे करना है
ये
वो आर्मी है
मेरे साथ बार-बार प्रोग्राम शुरू करते हैं लोग लेट आता है लगा ले मैं कैसे नहीं मेरे को अब तक सिस्टम कहां है मैंने बीच में राइट किया था राइट की तरफ था स्ट्रेट जा रहा था मैंने पहला किया लास्ट स्टेज आ रहा था यहाँ छापा गया वहाँ स्टेज राइट किया मैंने सिग्नल के आगे अच्छा वो सीधा उधर चला गया वो सेक्टर साथ ही
अरे वो ठीक है
सेक्टर बात है मैं तो सत्यम से बीकानेर तक चल के आया
सत्यम से
बीकानेर तक नहीं
मेरे 45 मिनट से पहुंचा
25 मिनट से भी कम है सुबह सुबह में दो लोग चल रहे थे वॉकिंग की अपने 50 हो
क्या
मर गए ये मतलब लेकिन सत्यम से चल के आना समझ गए इतना रात को 7 बजे अच्छा नहीं चला हां उधर बीकानेर आया मैंने स्ट्राइट दिया है और फिर वहां साइकिल से गए साइकिल में कल अभी लाइट लग गई है आपको अभी 2017 की जो आपको पता भी नहीं चलेगा बाद में तो मेरे को बाद में तो बीकानेर भाई मेरे को भाई कितना टाइम लगता है बस समझ में नहीं आ रहा है जो कितना दूर है वो चल रहा हूं मुझे आइडिया दिख रहा है वो उंगली वाला है उंगली है उंगली वो गाड़ी में हां सिलेक्ट होगा लोग सिलेक्ट उनको कर जाए खाली चलवी की तरफ है समझिए रोड है और आगे लेन में दो लोग चल रहे हैं इसके गाड़ी में ये आ गई गाड़ी तो इसमें क्या चीज ऐसा नहीं तो मैं भी उड़ा दूंगा पर नहीं छोड़ा ऊपर जेल में आएगा और बोले यार और नहीं चलो बड़ा ही चलो तो क्या
हाँ अरे मुझे हम लोग बैठ के पहले
कुछ नहीं
actually what about the other girl she's absent today also
Shreya
what we were doing which chapter we were doing this time economic importance for your economic importance sure complete with that and by the time Nevan I'll tell you what to do you without what I was discussing structure of bacteria are we done with that structure of the bacteria what was it look into that parts and functions parts of the bacteria and functions parts of the bacteria and functions Sadhya, I asked you to draw this diagram no? Bacteria look into that take the book one here check so the other day I asked you to
Look into the structure of the bacteria. देखो, what are the parts? Did you draw the other way diagram?
Yes, yes.
So now look into the parts of the bacteria. What are the parts of the bacteria? Let's start with that topic.
This chapter is over.
Wait,
In March.
I know that. So, what is your requirement below? What was this for you?
She had finished the scale. She has started scale and she has finished scale.
E not, ma'am. Is she your classmate? Your childhood friend? Your cousin? Younger sister? Elder sister? Which will know? So you have to address her as or sir. Good. So that means in the coming exam, see, not used to say that way, so she is finding it off. When you give respect to a person in the coming years, you will get respect. Follow? So that means in the coming exam, you are supposed to have skill. And movement locomotion. Yes. This is the other chapters, no? Movement locomotion is for you all also. No. Not for you all also, no? Asha. Now, for this group, it's over. For this group, movement locomotion is over. Skin also over. Skin also over. Okay, fine. And what?
Digestion.
Digestive system. Digestive system. That is done. Digestive system is done. Okay.
Respiratory system is yet to the
For your respiration.
respiratory system.
Respiratory system. A come for you. First of all, in this batch, three different absolutely different type of. Look at the chapter digestive system. Okay, now that chapter here it is over actually. We're already done long back. In the general lecture, this chapter is over. I can only discuss some questions with it because we want to give some questions. Explanation I have already done with them online. Explanation is already over for the chapter. And so you'll also do questions on the chapter, and in that process, if you have to ask anything, you can clarify. And then, so now for you, I'll start with this group first. Let us talk about the structure of the bacteria. What are the parts that are supposed to come across? A single cell. Okay, this is a single cell. It has got cytoplasm, a fair amount of cytoplasm it has. It has got cytoplasm can also be termed as the protoplasm. And then we talk about that it has got chromosome. The chromosome, what is the nature of the chromosome? The nature of the chromosome is a single, extensively coiled, long, thread-like structure. Single chromosome it has. Bacteria we are talking about, single chromosome it has. Does it have a cell wall? Yes, it has cell wall. But the cell wall, is it made up of cellulose for a bacteria? No, it is not made up of cellulose. Rather, the cell wall is made up of a substance known as peptidoglycan. You have to remember that word, peptidoglycan. Peptidoglycan, what is it? That's a cell wall substance of the bacteria. Cell wall substance of the bacteria known as peptidoglycan. Then what about the cell membrane? Does it have the cell membrane? Yes, it's a bacterial cell, a single cell organism. It also has got a cell membrane. Then when we look into the chromatin, I already told you what is the nature of the chromatin here? The nature of the chromatin is a single long thread-like structure, not like so many thread-like structures like organisms like human. Then when we talk about the cytoplasm, is this bacteria, first of all? Try to understand this bacteria basically comes out of the group Protista, if you remember 5 kilo classification, Protista. And these type of organisms, Protista, are either this type of organisms would be either autotrophic or heterotrophic. Would be either autotrophic or heterotrophic. So now this group of organisms may either contain chlorophyll, may either contain positron. By the time you read that part of the chapter, I'll come to you in there. Signed on? Now, next is we talk about what? The next part is what? We were talking about that chlorophyll. Does it have the chlorophyll? What is the mode of nutrition in case of the bacteria? The mode of nutrition in this group of organisms, Protista, can be either autotrophic or heterotrophic. but mostly they are heterotrophic. Few organisms, few types, few species of bacteria may also have the chlorophyll. So now that makes it autotrophic. Next is underline the word capsule. What is capsule? It's a protective cover for the bacterial cell. A protective cover for the bacterial cell. Do they have any locomotory structures? Yes, they have. What type of locomotory structures do they have? Flagella. So flagella is their locomotory structure. But what is the difference between the words cilia and flagella? So now that I talk about the bacteria, you are referring to the diagram. Now that I talk about the bacteria basically has a capsule and then the cytoplasmic structure and then a whole long thread-like structure. And then if you come across that it has a long wig-like structure, it has got long wig-like structures like this. Are we talking about the paramecium? Are we talking about the paramecium where there are fine thread-like structures and short and numerous? No. If it is something like this, then this is what we are talking about as cilia. What is cilia? Cilia would refer to small hair-like structures and many in number. But the moment I talk about flagella, what would flagella refer to? Flagella would refer to some long thread-like structures and majorly one or two, not more than that. Majorly one or two and long thread-like structures they are. So what is the locomotory structure majorly found in the bacteria? Flagella, not cilia. Next is we talk about the mode of nutrition. Just now we were also talking about the mode of nutrition can be autotrophic, but majorly heterotrophic. Underline some types. Heterotrophic also what types? Heterotrophic, there are 15 different types of heterotrophic nutrition found in the animal forms. What type? Heterotrophic for bacteria can be either parasitic or saprotrophic. Don't dream. Now saprotrophic and parasitic. So what is the meaning of parasitic? He is lost somewhere. स्कूल में आज स्कूल में कुछ हुआ क्या?
हाँ ना नाम ने
आज स्कूल ही नहीं था.
हाँ स्कूल ही था कोई इसको खड़ा किया था इसको नाम ने.
आज स्कूल ही नहीं था.
बाया नाम ने खड़ा किया था.
अरे आज स्कूल ही नहीं था. आज स्कूल ही नहीं था. तो ये लोग इतना झूठ क्यों बोल रहे हैं स्कूल का? हम झूठ लोग इसको नॉट बोल रहे हैं. कुछ तो नहीं करते. हम चाहते हैं बोल रहे हैं क्या बोल रहे हैं? हम कैसे बोल रहे हैं? बाकी जो बोले क्या Why were you dreaming? What are you dreaming about? Now, I should never ask this type of question. Too many questions at this particular way. Such a thing. You will have no embarrassment. You will put me into embarrassment. What like that? Not to ask.
देखो अब क्या बोल रहे हैं इतना बहुत भी नहीं
They want to ask. One. So now when we talk about types of nutrition, what type of nutrition? Bacteria basically follow heterotrophic. But what types of heterotrophic? Heterotrophic, just now I said, heterotrophic mode of nutrition can be again of different types. You stop smiling and stop breathing and concentrate. So now, what are the methods? Major methods of nutrition. You are asked questions. What is the mode of nutrition in bacteria? Are you supposed to write autotrophic? No, actually you are supposed to write heterotrophic. And what types of heterotrophism they show? Saprotrophic or parasite. Now what is the meaning of saprotrophic? Saprotrophic means when bacteria become a composite. And from the decomposed organic matter, they take the soluble into that is saprotrophic. How is parasitic different? For parasitic, or if it is a bacteria which is parasitic in nature, I would understand that this bacteria would be a type which enters into the body of a live organism and takes nourishment from that organism. If that is what? Then it is parasitic. But it is a dead animal or a plant, and the process of decomposition, it is bringing about the bacteria and then taking soluble nutrients, and it is saprotrophism. Respiration, when we are talking about the bacteria, tell me, what do you understand? Respiration in plants already over, no? Respiration in plants already over? Yeah? Respiration in plants, yeah. From that concept, if I ask you, bacteria, what do you remember? Bacteria is aerobic or anaerobic? Anaerobic respiration. Bacteria is a cell. Does it have adequate number of mitochondria? No, it does not. Mitochondria is not present in bacteria. So obviously the process of aerobic respiration cannot happen. And the process of anaerobic respiration does happen here. So now that we are talking about respiration, what type of respiration the bacteria is supposed to show? Anaerobic respiration. We'll talk about the reproduction and the process. The diagram all are important here. First of all, take 5 questions from this part of the chapter that I will be taking. Number 1. What is the cell wall of a bacteria made up of? What is the cell wall of a bacteria made up of? What is the cell wall of a bacteria made up of? Number 2, the words. Describe the chromatin material of a bacteria. Describe the chromatin material of a bacteria. And stop. Same question. Continue. What type of a cell it can be considered as? What type of a cell it can be considered as? What type of a cell it can be considered as? On the basis of nucleus only, huh? What type of a cell it can be considered as? Come to the next. What is the function of the capsule? What is the function of the capsule? What is the function of the capsule? Name the next question. Name the locomotory structure of bacteria. Name the locomotory structure of bacteria. Name the locomotory structure of bacteria. Full stop. Next part of the same question. What type of movement does it bring about? What type of movement does it bring about? What is the mode of next question? What are the 2 prominent modes of heterotrophic nutrition? What are the 2 prominent modes of heterotrophic nutrition? What are the 2 prominent modes of heterotrophic nutrition exhibited by bacteria? What are the 2 prominent modes of heterotrophic nutrition exhibited by bacteria? Next question. What is the prominent mode of respiration? What is the prominent mode of respiration? What is the prominent mode of respiration carried out by bacteria and why? What is the prominent mode of respiration carried out by bacteria and why? Okay, continue this. This is few questions we had done, no, the other day, Evan?
Yeah.
I gave you some questions.
What is the answer? What I would say it can be
Prokaryotic. Prokaryotic cell. Finish up, no talking. कुछ भी हुआ तो अनिश ना
हाँ भाई बोल रहा है
उसको लिख रहा है मामला
नहीं आपने सही समझा बहुत हौसला करते बेचारे की छोड़ दिया बहुत
क्या दुश्मनी है तुम लोग को इससे
हाँ देख देखो आपकी भी विश नहीं हो जाएगी उससे
हाँ तो नहीं क्या किया बता
देखो मैंने पढ़ा नहीं था तो मैं आपको शर्म आएगी सोचते मेरे एक स्टूडेंट है
ये बता तो बस
उसको भी अच्छा लगता है कि उसका टीचर
नहीं का
हम चेक कर लो कुछ नहीं है
उसको क्या लगता है
देख कितना बात तो सच बोलती है हाँ
एकदम प्लेन शर्ट पहन के तो आपके सामने वो बैठता भी है मैम मैं ही आगे बढ़ा थी इसके आगे इधर बढ़ा था ये बात कर रहे हैं कोई बात नहीं एक बार भी तो चेक करके साथ साथ उनको भी देखिए नो चलो फटाफट करो now
That is telling me I am instinct to see his shirt from that.
जो बड़ी चुगली करनी
I answer now, okay.
मैं ना बोलूँ
लोग स्कूल में पढ़ने जाते हो कि लड़ने जाते हो
Not bacteria, the prominent mode of respiration kind of bacteria is anaerobic. Ma'am, because it has less mitochondria or because it is killed if it exposed to it.
it has got no mitochondria in its cell it does not have mitochondria in its cell it does not possess mitochondria so cannot utilize the oxygen. It does not have mitochondria. At the same time, specialized structures to take in oxygen. That is what you write from this. Did you read the first part of the chapter? Anything that you want to clarify? Okay, where did you read? I'll give you questions only till that much. Acha, nahi aisa nahi. You have to read parts of the elementary canal first. How are you supposed to go through this chapter? How you should read? Digestive system hai. This digestive system is divided into elementary canal and digestive glands. Elementary canal and digestive glands. Okay, so this is first you should divide. So now that I talk about the alimentary canal, now you should know parts, functions of the alimentary canal first. So what are the parts of the alimentary canal? What are the parts of the alimentary canal? Mouth, that's okay, good. Stomach, small intestine, large intestine. Only these 5 parts you read once and tell me if there's anything that you want to clarify. I'll give you questions only from this one. Digestive glands, functions, later. Done? You finish writing down the answers? All of you? Okay, I will discuss with the answers right at that. You just leave some half a page, half a page. And then you will draw the diagram of reproduction in bacteria. Can you see that diagram? 3-4 pages of the diagram. Neatly, yes, my dear guy. Pencil. Make a start. And then proper diagrams. Make a habit. Before you go to the 10th standard, suddenly in one day you cannot be organized. अच्छा ड्रॉ करते हो बट पेन से करोगे तो उसका कोई मतलब ही नहीं है ना
मैम इधर के बात करेंगे
तो स्टार्ट करो
एक थैंक यू
क्या काम हो जाए बात है ये हम देख Neat diagram, proper label. Put the heading reproduction. That is the sexual method of reproduction in bacteria. We learn what is the method. Diagram पहले draw कर लो. Anyone any doubt in the answers? Tell me. हाँ करते रहो. I'll give you still many more are there. I'll give you. पहले ये part complete करो. I don't remember his name. Arnav. अच्छा Arnav, can you start now? Define peristalsis. आता है ये answer? क्या है? Define peristalsis. What is the answer? The rhythmic, लिखो. The rhythmic, RHYTHMIC. The rhythmic contraction and relaxation. The rhythmic contraction, and did you put the name of the chapter? Digestive system. The rhythmic contraction and relaxation of the walls of the alimentary canal. The walls of alimentary canal is known as peristalsis. Okay. Now go to the next one. One function of esophagus. Do you know what it is? What is the function of esophagus? Passage of the food. Passage of the food from mouth to stomach. Done, Spandan?
Yes, ma'am.
Now, did you complete the questions of the book? Or book? Or not? No, we're done this time. Now let us go back to this part. We are now to understand the process of sexual reproduction. Check the figure there. Process of sexual reproduction in bacteria. Now, in case of bacteria, though the process of sexual reproduction, we are saying there's nothing known as a male and a female actually. There's nothing known as male and female. It is only certain type of proteins which are formed different in two different types of cells. So that is it. This one by fission, reproduction by fission. I'm sorry, this is fission we are talking about. We'll also talk about conjugation. Conjugation is a sexual method of reproduction. First of all, we talk about the fission method, two methods by which, right now the first one. What are the two major methods of reproduction carried out by bacteria? So bacteria may carry out a sexual reproduction by fission and sexual reproduction by conjugation. A sexual reproduction by fission, a sexual reproduction by fission, and sexual reproduction is carried out by conjugation. So these are the two methods by which reproduction takes place in bacteria. So first of all, we talk about the asexual method. Now, in the asexual method, the major concept of asexual method by fission. What is fission? One cell dividing to form two cells. One binary. Same like binary fission that we know in Amoeba. So now here, what has to happen in order to bring about the concept of binary fission here again? What should happen? The parent cell, the primary cell, the parent cell should divide into two. But before that, the heavily material, chromosomes completely. So that is the simple process here that is also happening. So see the first stage what is happening there, the first parent cell has got a single chromatin, and almost in the shape of a circular, and a circular chromosome will undergo the process of duplication. That is what we get, which is the second stage, second figure. It is undergoing the process of duplication. Now, by the time the chromosome gets duplicated, what also will happen? The entire cell lengthens, almost same like amoeba. The cell now lengthens. As the cell now lengthens, what now next? As the cell is lengthening, what is the next step you get to see? The first stage is what? There is a constriction. Can you see there's a constriction in the membranes getting formed? There's a small constriction in the membrane. So the parent cell, first of all, what is the stage showing? If I have to show you the diagram here, first of all, this is a bacterial cell, and this bacterial cell has got a chromosome. What is the composition of the chromosome? DNA. Then what will happen? This will generally show process of duplication. This will generally show process of duplication. So now this is the duplicated DNA getting formed. उसके बाद जैसे ही duplication हो रहा है, what is the next stage? The entire cell starts lengthening. So now what will be the stage? The cell now starts lengthening. The cell now starts lengthening. So this is the parental chromosome, and this is the newly formed chromosome. But by that time, immediately we also come across that there's a small constriction getting formed in the membranes, a small constriction getting formed. And this constriction now slowly will start deepening. As this membrane constriction starts deepening, these two start getting separated. Absolutely, they get separated. Two new cells getting formed, the parent DNA in one, the duplicate DNA in the other. So that's a simple method of asexual reproduction. But if you have to talk about sexual method, then what? In case of sexual method, what happens? As I said just now, that in case of the sexual method, what is the name of that sexual method? The name of the sexual method is conjugation, but there is nothing known as a male and a female. Then in that case, what should be the method of division? This is a parent cell, say suppose, and it has got its chromosome. This is one parent cell. Then what would be happening in this parent cell? This parent cell will generally form a passage with the other cell, resulting in the formation of a passage pathway, and then some duplication will happen, and this duplicated DNA now slowly will start migrating and start entering into the next cell. So if this is the method which is formed by making contact, two cells are making contact, there is nothing known as the male female, but only they are termed as the positive strain and the negative strain. Only because they secrete different type of proteins. This is the method, this is their sexual method of reproduction. So you can write down the sexual method, the diagrams are not necessary for you. Sexual method of reproduction is carried out by conjugation. Sexual method of reproduction is carried out by conjugation. Sexual method of reproduction carried out by conjugation. Now, as the method of reproduction happens in the bacteria, it is also possible that now the new bacterial cell is getting formed, but the environmental conditions are not suitable. The environmental conditions are odd. The environmental conditions, the temperature, atmosphere. What happened on our next question? Did you finish? One function of esophagus done. Dental formula would be right. Adolescent to the adult. Third answer kya hoga? Which type of teeth is absent in the infants? Which type of teeth? Which type of teeth? Now, with reference to this, adolescents, you know what is a dental formula. Adults, you know what is the dental formula. What is the dental formula you have written for adolescent?
2, 1, 2, upon 2, 1, 2 upon 2 is equal to 28,000.
2, 1.
2 upon 2, 1, 2.
2, 1, 2. No, so we say dental formula is 2, 1, 2, 2. So 2, 1, 2, 2 means upper jaw, lower jaw will be the same. So 2, 1, 2, 2. So which type of, what is the difference? 2, 1, 2, 2, adolescent and adult should be 2, 1?
2, 3.
2, 3. Now if I have to write down for an infant, what should I write? 2, 1?
0, 2.
0, 2. So which type of teeth is absent now? Huh? Premolars. Premolars are absent. So remember that. Premolar cells. Okay.
I have a doubt. Question number 5.
Fungi are saprophytic. उसमें saprotrophic होगा. Same saprotrophic है उसमें. Answer. Saprophytic. Yesterday I told you. Saprophytic meaning fungi. Where do we find? How do we find fungi around the green and the dry surface? Where do we find fungi when the surface is damp? And at all when the process of bread is getting stale, food is getting stale, rotted, fruit, then only we come up. Then what is meaning? The process of decomposition where fungi is there. No. So now we understand what is the definition of to write. Fungi are saprophytic. Give reason. Write down. Write down. Fungi can absorb. Because say the function of incisors. It's okay. What is the function of incisor? Will you be able to write? These are straight answers already there in the book. Fungi absorb soluble nutrients from decomposing organic matter. From decomposing organic matter. From decomposing organic matter. Good stop. They grow best in damp and decomposing surfaces. They grow best on the damp and decomposing surfaces, hence known as saprophytic. Hence known as saprophytic. Next, you will be able to. Now, next is this side. When we talk about, I was saying that when this bacteria up here,
खुद को लेके हंस रहा है.
तुम लोग हंस रहे हो. अच्छा, these I understand. You 3 months of details. We need to be right. When we talk about this, when the new bacterial cell are getting formed, a lot of bacterial cells are getting formed. Now what will happen? Sometimes it is also possible that the environmental conditions are not suitable. So now what? This bacteria, new bacteria starts forming protective coat around them, protective structures around them. And this new bacteria, the new daughter cells, are covered by some protective coat. Can you see that? The spore, the diagram of that spore, can you see a bacterium forming a spore inside itself? So what is happening is, this entire bacterial wall, the entire bacterial cell starts resulting in the formation of a structure known as a spore, where the nuclear region remains protected. The nuclear region remains protected by an extra cover. But is this bacteria ready to survive in the external environment? Not yet. Now when the conditions are suitable, what will happen? This entire outer cover will burst up and the new bacterial cell will come out. So is spore formation a method of reproduction for the bacteria? No, it is a structure formed to avoid unfavorable conditions. Because spore formation is a method of reproduction for fungus. Is it also the same for bacteria? No, spore formation is not a method of reproduction. It is just a mode to protect from undesirable, unfavorable conditions. Now, why are spores formed, or why should spore formation happen? Take those points to remember. Spores are usually the resting stages which can withstand environmental conditions, such as they can tolerate extreme dryness. Can you see those points? Extreme dryness. Why spores will be formed? When does spores get formed? When the environmental condition is extremely dry. I told you unfavorable condition. Number 2, quite a number of times. Say suppose the temperature becomes extremely unfavorable, too high, too low. At that time, also the spores will survive. That's a reason we come across. The bacterial spores or the bacterial infection is also so fast because the spores do not get easily destroyed. Third is they can also tolerate a whole lot of poisonous chemicals also. They feel that whenever there's a bacterial infection, just add or put some antiseptics or harsh chemicals, and then the bacterial infection on our skin or any other part of the body that will not be healed. Unless and until we take specific medicines or antibiotics for them, then that will not be cured. Next, if we talk about the useful role of the bacteria. This is very important. Useful role of the bacteria. What is the useful role? Number one, antibiotics. What are antibiotics? Do you know? Antibiotics क्या है?
And they're produced in the body. नहीं? Antibiotics are when they are.
Did you take antibiotics?
Yes, no, of course.
बहुत सारा. And when did you take an antibiotic?
I'm जब कोई extreme level पे वो हुआ था. Like there are antibiotics for everything. So definition one need to take example.
अच्छा exam many questions can answer they were here. No, that is vaccine. वो तो vaccine है. Antibiotic कब लिया था? क्यों लेते हैं हम लोग antibiotic? When did you take antibiotic?
The spreading of bacteria.
Antibiotics that we don't have in the body. Antibiotics.
Antibiotics have
Because that is a supplement for antibiotics that we can't create.
And generally जब कोई
Antibiotics the body cannot make. Antibodies our body can make. Antibiotics, that's why I asked you. Antibiotics and antibiotic close terms. So that concept is absolutely different. When I talk about antibiotics, remember these are extracts of. Actually, these are extracts of bacteria or virus, which bacteria or fungi, which can kill other bacterial organisms. So long time back, when we talk about how did the concept of antibiotic discover? How did it get discovered? Fleming, very famous biologist, scientist, Alexander Fleming. So what he was doing, he was in his laboratory, he was working with certain type of bacteria, Staphylococcus. So he was working with this bacteria. He was trying to make clusters of bacteria, generations of bacteria. But every time he was observing, जो bacterial plate है, after some few hours or some couple of days, what was happening? ये जो bacterial plate full हो जाना चाहिए था with bacteria, that was not happening. Rather, bacterial plate में छोटे-छोटे patches हो जा रहे थे and clear areas हो रहा. So the bacterial plate, which should have been full with clusters of bacteria, that was not happening. Rather, the entire bacterial plate, when he analyzed, he saw some clear areas. Then he initially felt that that was a mistake happening. He again repeats the experiment. He again does it. Again the same thing, right? A number of times the same thing happened. Then when this happened, say suppose now I talk about a bacterial plate. So what was he observing? Some clear areas, and उसके आसपास बहुत सारे bacteria that he tried to grow. So ये जो bacteria के आसपास जो clear areas हो रहा था, why those clear areas? Then he started analyzing the chemical substances, and then he started analyzing, understanding, and that also we are talking long back. Alexander Fleming was not there a decade back. We are talking long back. Today, whatever type of sophisticated microscopes are there, it was not there at that time, no? But still, he tried analyzing what type of organisms, so is it something here? So he tried taking substances from here and observing his microscope, and then he saw some certain additional, some new type of organisms were there, which are not similar like this. He saw these are new type of organisms, not similar like this, and then he started analyzing what type of chemical substances they must be producing, and then he started separating them, and then segregating them and filtering them, and all these processes he did. And ultimately he found that there are some type of organisms, and these type of organisms were releasing some type of chemicals. They were releasing some type of chemicals, and these chemicals were responsible for killing the bacteria that he was trying to grow. So after a couple of years, he could understand that this type of bacteria, this type of organisms are a type of a fungus, and these type of organisms were later identified as Penicillium notata. Penicillium, a particular species of fungus, Penicillium notata. So when we are talking about penicillium is one type of a fungus that he found. Underline that word, underline the name of the organism. Penicillium. So penicillium notatum, also Penicillium chrysogenum, another type of species. Penicillium notatum was easily identified. So this Penicillium notatum, what happened? Could you see? है ना bracket में देखो, देखा? Penicillium notatum, Penicillium chrysogenum. These are the two species that you could identify. And then he found that the chemical substances released by these had an effect of killing other microbes. Had an effect of killing other microbes. Then what was the name given to these chemicals? The name given to these chemicals was penicillin. And these penicillin was now tested for a variety of other microbes, and it was found that talk about a plate of some bacteria and put this chemical substances here, the bacteria gets killed. The bacteria is died, and the bacteria cannot multiply any further. So this was tried on a variety of other organisms, variety of these microbes, and every time it was found to be successful. Then this was tried on human, and that was a miracle where in the field of medical science that proved to be a boon where so many different type of infections, so many different type of bacteria could be killed. So this penicillin remained an ideal antibiotic for a large number of years, for a long period, almost for about 2-3 decades. It remained a dominating antibiotic. So what is the definition of an antibiotic then? What is an antibiotic? Antibiotic is actually a chemical substance or a chemical extract from a bacteria or a fungus, which can destroy other bacteria, other microbes. But today the antibiotic that we take for a variety of infections, today the antibiotic that we take in for the skin infection, eye infection, etc., are these also same way made from the fungus or the bacteria? No, today not. Today these are just chemical compositions. Because extracting the chemical from a fungus or the bacteria is a strenuous process. And today with the alarming increase in population, not possible at all. So today the antibiotic that we are advised to take are not at all bacterial or fungal extract, the base is the chemical compositions. Okay. Then what is the first antibiotic discovered? Penicillin. Which year? 1929. Who did that? Lemon. Which was the organism on which was the organism? Which was the or so which was the two types of organisms that we have noticed releasing the chemical substances. And Penicillium chrysogenum was actually identified as notatum later.
Yes, sir. Commonly it was
Was identified, but notatum is actually the common name. We usually call it as notatum. Penicillium notatum is a more common name.
we've seen it in the next two of the previous.
Notatum. Notatum is a common one. Then after that, what was the second antibiotic discovered? That was streptomycin. And today so many, so in a row, so many were discovered. Then here you have to understand that what are some of the characteristics of a good antibiotic? Then it should be a good antibiotic. Now, penicillin was a good antibiotic. I said it was a dominating antibiotic for almost few decades. Today, do we get penicillin in the market? No. Do you remember right from the time you were an infant, or right now for the last 5 years also? Penicillin you get in the market? No, nowadays in no pharmacy will get penicillin available. But I don't know. I mean, if you also remember last 5 years, you remember, did any doctor prescribe penicillin? No. Penicillin was prescribed long back, but later it was identified that penicillin had a lot of side effects also. Now, what are some of the characteristics of a good antibiotic? What is a good antibiotic? What was the reason that penicillin became obsolete nowadays? Because when we talk about penicillin at one particular period of time, yes, it was curing certain type of infection, but it was giving adequate amount of side effects also to the human body. Some adequate side effects also were there. What were some of the side effects? Some unconsciousness was also there. Some digestive problems was also there, etc. Headache was there. Dehydration was there. So all these. So now I said that nowadays the antibiotic that we consume are basic chemical compositions which can clear human bacterial infection. And now with so many compositions coming out, it was found that there are so many such type of composition which can treat more than one infection. at the same time, but with minimum side effects. So today, if you remember the last time antibiotic that you took, maybe for your throat infection, for your fevers, for your stomach infection, hardly it must have given you any side effects. Hardly. Today, if you remember, or if you have heard about from your grandparents, antibiotic, they would generally be scared of taking antibiotics because for them antibiotic is something scary. Why? Because when they had taken antibiotics, the experience was with a lot of side effects. But today, I think we would prefer more to take antibiotics because infection is gone very fast and we have to constantly keep up. Yes.
Antibiotics.
But comparatively much lesser.
If somebody does have antibiotics, does have effects. It should have.
It should have, correct. Everyone is correct. It should have. But at the same time, if you understand, as I try to put it very simple, if you ask your grandmother, she will always say, no, antibiotics are not good because they create a whole lot of, because today's antibiotic and that time's antibiotic, whole lot of difference. But today, you would always prefer to take some antibiotic in the morning and go for a walk. And that will be okay. It doesn't, we don't feel also. So not that much of a side effect as today's antibiotic. But the previous day's antibiotic, if you take, you have to really lie down on the bed. You cannot go out for work because it really creates a lot of disturbance in the body functions. So today the antibiotic is made in such a way bacterial infection is also killed and body metabolism also does not suffer. But yes, in certain individual doctors when they prescribe, they tell you also that it may create some minor headache or stomach trouble, etc., may happen. But in some people, it may not happen anything. For example, when I take antibiotic, nothing happens. So, but yes, in some people it happens. Body to body, the difference is there. So that is what. So here, the antibiotic, what should we think that? What is some of the criteria, characteristics of a good antibiotic? And I done what? Number one, it should be able to kill a variety of organisms. Again, here today, if you see the prescription for the last 2 years, 3 years, you may come across, if you have one common doctor, you may see that the doctor must have given you the same antibiotic for your throat infection also, and maybe for your skin infection also, and maybe for your indigestion also. Doctor the same antibiotic there, but it worked. It worked. Why? Because actually, if it is a good antibiotic, it should be able to treat a variety of infections. If it is a good antibiotic, an effective antibiotic should be able to treat more than one type of infection, then it is a good one. Then what is the other criteria for a good antibiotic? Less side effects. It should be having less side effects. See the third point? It should not kill the good bacteria of our body. Are there any good bacteria in our body?
Yes.
What does it do?
It acts as a, like a soldier. It prevents the entry of other bacteria, like a
At the same time, there are a whole lot of bacteria in our intestine which also enhances the process of digestion. There are a whole lot of bacteria in small intestine. Right now, if you take out a small amount of your saliva, put it under focus, hundreds of bacteria. So this bacteria in our stomach, in our mouth, are actually enhancing the functions when carried out in those parts of the body. But these are good bacteria. And if these bacteria are killed by consuming an antibiotic, then that's a side effect. And that's the reason we will suffer from other effects. Other uses of antibiotic, what am I supposed to write? Other uses, quite a number of times, the antibiotic should also be acting as a feed, a nutritive feed. Antibiotic should also be acting as a nutritive feed for treating animal feed. So that's the most important point that I should remember. Treating animal feed. We'll take a break and then continue again. Did you finish with all these questions? Do you have any doubt here?
Hello.
What is wisdom to the good, right? Or one difference in the crown and the root. What do you know there? Crown and the root?
It is the hardest substance.
Crown. Crown.
It consists of 11 and 10.
So difference can be okay. Crown and root. I asked you one difference to write. One difference to write. Crown and root. What difference will you write?
Consists of 11 and 10 and root. It consists of jawbone, cement, pulp, and vein of blood.
सबसे इम्पोर्टेंट डिफरेंस ये निकला है क्राउन इज द पार्ट ऑफ द टूथ अबव द गम रूट इज द पार्ट ऑफ द टूथ below the gum. That's the main difference, right? That's a major distinguishing feature, right? Apart from that, the conversation. Okay, can you take a break?
Wow, Alice को लगता है देख कितना मेरा अच्छा ये फोन रहा है मेरे फोन भी है
हाँ
सब मेरे से
मेरा जी था
थैंक यू अरे क्या है
अनिश पे मतलब किसी को दे तो पूछ रहे थे कि
हाँ
क्या होता है नहीं कोई तो शुगर केन के बारे में बोल रहा था
अरे यार
तो फिर कोई तो बोलते हैं कि शुगर केन का फॉर्मूला भी होता है तो अनिश क्या बोलते हैं शुगर केन का फॉर्मूला मुझे सिंपली एस्पेल बोल देता है
अगर उसके अंदर शुगर है तो बॉडी बॉडी अंदर जाना क्या ग्लूकोज शुगर मतलब अबे लास्ट में ग्लूकोज ही बनना है कौन सा भी शुगर रहते लास्ट में ग्लूकोज ही बनना है अच्छा ठीक है
हाँ समय में प्रॉब्लम छोला भी नहीं है बहुत छोटा पार्ट रहेगा कुछ नहीं हम लोग तो चेहरा छोला खाई नहीं है
अरे मॉडल ऑफ देखा है जर्मनी जापान
वो वाला सिस्टम
ना लॉस के लिए देखा आज वापस आ गए आज मैक्सिको करना सनवार ना आज का ये चक्कर है हो गया गांव कोई अच्छा अर्जेंटीना मेक्सिको का कीपर है
वही देखने था मैं फ्रांस जीत गई
अच्छा जी
कुछ तो बने रहेगा
हार्ट फेल हो जाएगा
मैंने सोचा
मैं वो लड़की फ्राई नहीं देती
फिर पता है मेरे को नहीं मालूम क्या पॉइंट क्या करना है आज के कौन सा कर रहे हैं क्या भी
अब इधर मेरे हाथ से देख
अरे फर्क नहीं पड़ता बोलेगा Wah, ini kacau ini. Ada tuh dia kan itu. Apa tadi? Dia bilang, "Oh, bisa ini keren banget." Apa mereka suruh itu? Ini apa ya? Mana? Bukan aku mama saya. Jadi itu dah kualitas kita wakil pada. Jadi juga kendalanya ada. Berani kan? Mana ya?
Jangan. Kita.
Paling lama dia untuk kandidat ini. Monday को छुट्टी है मेरे को पता है कि हमारा इंग्लिश कोर्स और मान लीजिए ट्यूसडे से मेरे को वारना को बोले जरूरत नहीं अभी लोग तेरे को बात नहीं हम लोग आफ्टर लॉकडाउन उन्होंने क्या किया एक ग्रुप amount of बच्चे ठीक है
only if but it's not very color.
Sit down, come this side, I will come this side and
अब देखा बड़ी चिंता बनती है ये टीचर ने वो करना है क्या ये देखो बस नहीं करो तुमको भी बात नहीं है
नहीं ना आधे नहीं करो
I take some questions from there. What are antibiotics? I give you again. Read. I gave you questions only till esophagus. Now I give you questions on stomach, small intestine, stomach, small intestine. Here I gave you questions. I think only till fungi, plants, animals. Okay. Now for invertebrates, I'll give you questions for invertebrates and vertebrates. Check. Plantain, only the names you should know with one example. Find that happen by one example. And invertebrate, vertebrate, I'll give you some questions. This group continue. The first one, what are antibiotics? What are antibiotics? What are antibiotics? Give two examples. Give two examples. Next question. Name the first antibiotic produced from a fungus. Name the first antibiotic produced from a fungus. Name the first antibiotic produced from a fungus. Next question. Name the biologist. Name the biologist who introduced the first antibiotic. Name the biologist who introduced the first antibiotic and in which year? And in which year? Name the biologist who discovered streptomycin as an antibiotic. Name the biologist who discovered, his name is Simon Roxburgh, his name. Name the biologist who discovered streptomycin as an antibiotic. Discovered streptomycin as an antibiotic. Next part of the same question. Name the organism from which the antibiotic was discovered. Or name the organism from which the antibiotic was obtained. Antibiotic was obtained. Next. Write one difference between antibiotic and antitoxin. Write one difference between antibiotic and antitoxins. One difference between antibiotic and antitoxins. Next question. What is the role of steroid? What is the role of 7? What is the role of 7? Question mark. How does it? How is it used? How is it used in? How is it used in treating? In treating different type of infection? Try the first part of the question. The first few questions will be able to answer. The last two I will discuss and then you can write.
क्या कर रहे हैं?
क्या हुआ?
आप भूल ही नहीं रहे, नीरज जब मैंने बिना देखे लिखे वो भी सही है, और फिर नीरज देख के लिखे दो में उसको देखो मैं देख के नहीं रहा।
चार चौदह आंसर
क्वेश्चन बोल लिखा है
वो क्वेश्चन 4 का आंसर है। आया राइट में दिमाग से कितना
You all like the same second, no? Same second.
हाँ
which is not a last
Ma'am was telling that you should have discovered some practical if there is maths in every day, 365 days maths, so that becomes the end of school.
exactly
You think you go to school only for math?
no there is no doubt
tennis court
So take it and
अब AC है मेरी रूम
क्या अच्छा निकाल भी जिसका binomial nomenclature है ना binomial nomenclature ना
हाँ मुझे पता है
तुम अपना काम करो
in which the first part has the name of the general system
I don't know what to tell you yeah इतना कुछ नहीं लिखा
वो जाएगा नाम देने फाइन नाम
हमारे बस एक
those days I'm now to washroom मुझे क्या करना है मैं जाऊँ I'm now to washroom
वही ना मुझे तुम्हारा
हाँ 4 क्वेश्चन लिखिए अभी लास्ट में
आज मैंने जा रही थी
आप चेक कर लीजिए
fast fast अच्छा binomial nomenclature I'll tell you what to write exactly and how to do some examples.
very sophisticated I'm not getting to the getting this answer now. One difference between anti-coordinate.
I don't think the last two I'm going to give. इसके आगे ये मैंने ऐसा किया है Then
Yes, ma'am.
Sure. What is this? What are the questions I give you? I gave you question, the first one, could you write?
Water, antibiotic, and medicine.
What are antibiotics? What are antibiotics you wrote?
The antibiotic is a chemical substance produced by a living organism which does not
Antibiotics are chemical substances produced by organisms which can produced by bacteria or fungus. Basically, these are organisms. These are chemical substances produced by bacteria or fungus. Okay. What is the next one? I think I asked you write examples. Two examples.
streptomycin, chloromycin.
Penicillin. Any two that also you can write penicillin, streptomycin, chloromycin, any of this two. Third, next question.
Name the first antibiotic produced from a fungus.
That you know. The next question.
Name the biologist who introduced the first antibiotic and which year.
Alexander. Okay, that also you know.
Name the biologist who discovered streptomycin as an antibiotic. Name the organism from
That
which.
also you have written correct. Next.
Write one difference between antibiotic and antibiotic.
Now we talk about antitoxins. What are antitoxins? So antibiotics, what if we come across, these are bacterial or extracts of a fungus, which are processed, filtered, and then when it is introduced to the human body or other animal body, it can cure bacterial infection.
Now ये chapter में हम लोग सिर्फ mark parts हैं जो mark किए हुए.
So maybe it is a marked part of yeah.
मैम वो ब्लू बॉक्स करना है उसके बाद वैक्सीन वैक्सीनेशन
All the three definitions are important. Yeah, I'm talking about those only. So the next is when we talk about the antitoxins. What are antitoxins? Antitoxins are say suppose antitoxins are actually chemical substances which are released by the WBC. So when I talk about the antitoxins, are the antibodies, are the antibiotics released by our WBCs? No, antibiotics are not released by WBCs. Antibiotics we are taking from outside. But what is an antitoxin? It is a chemical substance which is released by one type of WBC itself. But what will this do? Quite often, we may come across and we get bitten by a poisonous insect. We get bitten by some other different type of organisms, harmful organisms. So now these poisonous chemical secretions, whenever enter our body, these chemical secretions also need to be neutralized. These chemical secretions also need to be nullified, otherwise they may destroy cells and improve the body. So how is this? How are these poisonous substances entering? Now, suppose we talk about a bee sting. This bee, whenever it stings, a chemical substance enters our body which is really poisonous, which can destroy certain tissues of our body. But now inside our blood, we already have the antitoxins, despite of diseases. So these antitoxins can neutralize poisonous chemical substances that can enter our body. Antitoxins are produced by our body. Are antibiotics produced by our body? No, they are not produced by our body. Can antibiotics destroy harmful chemical substances. No, they can destroy germs, a full germ they can destroy. Antibiotics can destroy the entire bacteria, but antitoxins will destroy only the chemical substance released by bacteria. The bee stings, the snake venom, says that. Now these chemical substances, when it is entering, the antitoxin will take care. But if I am talking about a germ entering, a bacteria entering, the antitoxin cannot do anything. Let us write down the difference there. Make a column, write two differences, antibiotic, antitoxic. The first point you can write antibiotics. Antibiotics are introduced. Antibiotics are introduced into the human body. Antibiotics are introduced into the human body. Antitoxins are produced by WBCs in the human body. Antitoxins are produced by WBCs in the human body. Antitoxins produced by WBCs in the human body. Number 2. Number 2. Antibiotics under that number 2. Antibiotics. What did you come here? Antibiotics. Second point, देखो. These are chemical substances that can destroy microbes. Chemical substances that can destroy microbes. Antitoxins. Chemical substances. Antitoxins. Chemical substances that can neutralize. Chemical substances that can neutralize harmful chemical or that can neutralize poisonous chemicals, that can neutralize poisonous chemicals that enter our body, that can neutralize poisonous chemicals that can enter our body. Poisonous chemicals that enter our body. Now, what is the next question?
What is the role of cell? How it is in doing different types of information?
Now, see that there is one more word, toxoids. Can you see toxoids? So, what are toxoids? When we are talking about these different ways of toxoids are actually when we are talking about these are different ways of prevention. Also, we can say toxoid is another one. So, we are talking about toxoids are what? Toxoids are inactivated toxins. Any type of a toxin released by one type of an organism. If it is introduced in our body in a inactivated form, it may not have any effect on our body. But inactivated how? Treated with chemicals or by heat. In an inactivated form, if it all can be introduced. From that, the definition is a small concept there. Inactivated toxins of certain bacteria that can stimulate production of certain antibodies. Now, just here, actually, the concept of toxoids to explain is a big thing. But it's very simple, try to understand. Whenever there is a chemical substance entering our body which is not of our body, or a cell entering our body which is not of our body, WBC is a type of a cell which can easily understand. It is something like a security guard. Any chemical entering our body, not of our body, any cell entering our body, not of our body, that's the first type of cell which can sense that something foreign is entering. Something foreign is entering. So whenever there's toxoids, what are toxoids? Toxoids are basically chemical substances which can be released to inactivate any type of toxins also that are entering in our body. So now, since we might talk about any type of a chemical substance released by a specific type of an insect, poisonous chemical substance released by an insect. This is inactivated also. We inactivated also that. After that, we are entering. After that, we are allowing that to enter our body. Toxin which is inactivated. the toxin which is inactivated, अगले दिन से ना एक टेप लाना, पेंसिल को बांध के रखना, हर दिन के लिए. So now, when we are talking about ये जो toxin, what are toxins? Any type of a chemical substance released by an animal or an insect, we make it less poisonous, inactivate करते हैं, inactivated toxins are known as toxoid. But even then, it is already inactivated. Is it going to harm our body? No. But we still take it inside our body, inoculate, injection, में, oral, whatever. Take it inside. Is it going to give rise to anything, any reaction inside our body? Is it going to give rise to any reaction in our body? Yes, again what I said, WBC releases, they understand that these are secretions entering our body, but it is not produced by our own body. It is not of our own body. Then we give the type of cell which start producing something, WBC. WBC will start producing what? Antibodies. WBC start producing antibodies. So what is the definition? Let us write down the definition. What are toxoids? What are the toxoids? Toxoids are inactivated toxins. What are toxoids? Right now. What are toxoids? Toxoids are inactivated toxins. Toxoids are inactivated toxins. Toxoids are inactivated toxins which can stimulate production of antibodies inside the human body, which can stimulate production of antibodies inside the human body. Inside the human body. Full stop. Example, toxoids are used in treating diphtheria, tetanus. 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 Now, so that is the concept of toxoids. Any type of a harmful chemical substance we treat it first, then we put inside the body, then also the. Now, this is the same concept again when I talk about serum and to some extent vaccines. But let us talk about serum. What is happening? In case of serum, first of all, let us learn one example here. Okay. You are done with these questions? Now, here, say suppose I talk about the concept of serum. What is this? I look into one type of a virus, and this is known as the cowpox virus. This cowpox virus is quite similar to the chickenpox virus. So this cowpox virus is introduced into the body of the cattle. Why? To actually prepare serum. Now, what is the meaning of serum? So now this cowpox virus is introduced into the body of the cattle. Now this is a live germ. This live germ is introduced into the body of the cattle. Is this cattle body going to suffer from any symptoms? Yes. The cattle body will have some blisters and some secretions from the respiratory tract. Mild symptoms, but symptoms of chickenpox is quite severe when we talk about the symptoms of chickenpox. No. So here, what are we trying to make? We are trying to make some medicines for chickenpox, to make it very simple. So what are we doing? Chickenpox, a related virus we take, cowpox virus introduced into the body of the cattle. The moment it is introduced in the body of the cattle, what will happen in the body of the cattle? The WBC starts releasing antibodies. And where is these antibodies present? In the plasma of the cattle. The antibodies will be produced by the WBCs, and where will these antibodies be now released? Where will it be present? In the plasma. So now what is the next step? What do? We collect a small portion of the plasma. So from the cattle body, what are we doing? We are taking some blood samples. Once this cowpox virus is introduced, we wait for a period of a week, allow adequate amount of this antibody to be produced, and then we'll take some blood samples from the body of the cattle. This blood sample From the blood, the plasma has to be filtered out. The cells and all these things we don't need. We need only the plasma. So this plasma, which contains the dissolved antibodies, which will help in curing a related type of an infection, that is what is known as a serum. So what is this serum? Serum is actually the blood plasma, which contains some of the other type of antibodies. So now this plasma contains antibodies for cowpox virus. So this serum, when introduced into the human body, can it give a little bit of an immunity? Can it give immunity for the chickenpox virus? Yes, it can. Yes, it can. So that is what we are talking about as serum. Is the concept same like vaccines? No. In the case of vaccines, are we talking plasma in the case of vaccines? No. In case of vaccines, what are we doing? We are just taking a suspension of some dead germs and then introducing inside the body. But the function is same. When these dead germs in a suspension entering inside our body, the WBC is doing the same job, release antibodies for that germ. So now after 6 months, if the live germ enters, this antibody is already there in the plasma. Are we suffering from the symptom? We are not suffering from the symptom. Are these two terms clear? Serum and vaccines, are these two terms clear? Echo it out. You read these two paragraphs and I'll give you questions. This is important. Serum and vaccines. Vaccines, you should know. What is binomial nomenclature? Karo? I got all of you this side. Yes, vaccine ka paragraph and yes, serum ka paragraph. Serum. Just read. I explained you just now. I'll give you certain questions on this. What I'm feeling now. What is binomial nomenclature? The process of naming organisms. Process of naming organisms using two names. Using two names, put a hyphen. A generic name, a generic, and a specific name. A generic name and a specific name is known as binomial nomenclature. Can you see some examples of scientific names in your book? No. That's the common name. That is not the scientific name. You can write Panthera leo, you can write Homo sapien. Okay. Write down. This side all done till here. Did you write down this properly? What you wrote? 2.0 or 1.0? 1.0. Function of mucus, could you write? You could write the function of mucus. Next is you complete the questions, some of these, then when there's a lecture for explanation I will do later. First, now you'll have to complete the questions only. What is the significance of the extensively coiled, long coiled small intestine? What are you supposed to write? What is the significance that the small intestine is extensively long and coiled? So that, no, what is the significance that the small intestine is extensively long and coiled? You are supposed to write. The partially digested food passes through the small intestine slowly, in about efficient process of digestion. So that the partially digested food can move through the small intestine slowly. The movement should be slow, so that the enzymes can act on the partially digested food and bring about the process of digestion efficiently. Will you be able to write? Now this side, what I told you to write. Okay, I asked you to go through now. So let us now write down differences between the serum and vaccine. Let us write. What is a serum? How is it different from vaccine? What is a serum? How is it different from vaccine? What is a serum? How is it different from vaccine? The next class we will take the goal of taking. Let's see. What is serum? How is it different from vaccine? देखो.
I'm not sure calling.
No. How is it different from vaccine? Serum is the blood plasma. Serum is the blood plasma. Serum is the blood plasma from which now that's a typical definition. Actually, we say that serum is the blood plasma from which fibrinogen is removed. But here the concept of fibrinogen is not necessary. We are not talking about blood clotting or those factors right now. Serum is the blood plasma from which fibrinogen is removed. From which fibrinogen is a protein. From which fibrinogen is removed. And contains antibodies and contains antibodies produced and contains antibodies produced by the WBCs. Full stop. Next sentence. This blood plasma can be used for treating. This blood plasma can be used for treating various infections. Can be used for treating various infections. Or various infections or conditions, treating various infections or condition such as snake bite. So here we are talking about two conditions. Let's say serum. What is basically serum? Serum is a plasma. Serum is a chemical component of the blood itself, plasma. And the plasma containing either the antibodies or it may also contain some antitoxins. Now I talk about a person who is already bitten by a snake. The person who is already bitten by a snake, the blood has got already introduced to the venom. Now, this venom is neutralized with certain type of chemicals. The person is saved. So what now to do? From this person who is already treated for the snake bite, take out the plasma from his body. His plasma already made, first of all, take out the blood and then separate the corpuscles and the plasma. Now the plasma of this person can be. Now this person's plasma is what is known as serum. This serum has got what? Antitoxins for the snake bite, antitoxins for the venom, anti-venin. So this person's plasma, also known as serum, is containing the anti-venin. So this person's plasma, can it be given to another person when the person suffers from another person suffering from a snake bite? Yes, it can be. So remember these two concepts. I gave you two examples here. एक तो है या तो viral infection leads to a related secondary infection, or also for snake bite, conditions of snake bite. Achha. Now let us talk about vaccine. Have a side, right? Vaccines are suspensions of Vaccines are suspensions of. Suspension means it's a preparation. Vaccines are suspension of weak or dead germs. Weak or dead germs.
I'm not getting that answer 6.
Number 6. Mention the specialized region of small intestine. The small intestine, the ileum of the small intestine has numerous finger-like projections known as villi. The villi increase the surface area for the process of absorption. 2 points लिखना है, दो sentences लिखना है. What I said, vaccines are or dead or kill germs. Weak or dead germs, I said, no? Weak or dead germs, which when introduced into the body, stimulates WBC, stimulates WBCs to release antibodies, full stop. To release antibodies, full stop. Done. To release antibodies, full stop. This provides immunity against. This provides immunity against the specific. This provides immunity against the specific. This provides immunity against the specific germ. Now tell me if you have understood serum and antibody. Vaccine, I'm sorry. Serum and vaccine. Serum, should it be taken? Can I take the serum much again? The person has not suffered snake bite. Can you take the serum? 6 months that's something as serum. No. When the condition has happened, then only. The person is bitten by snake, then only the antivenin to be given. Can the person take the antivenin 1 year back? Will the effect be there? No, the effect will not be there. So it has to be taken. That means the serum has to be taken always at the time of the condition, either the infection or the snake bite. But what about the vaccine? Can we take the should we take the vaccine at the time of infection? We can take it very ahead of time. We can take it very ahead of time. So now that we take the vaccine today, we are immune for a long period of time because these antibodies will not get excreted out. They are not released out from our body. They will remain in the blood for many, many years. Then this you write. Name the categories. Next question. Name the 2 main categories of vaccines. Name the 2 main categories of vaccines with 1 example in each case. Name the 2 main categories of vaccines with 1 example in each case. Here we go. 2 main categories. Now, so the 4 categories of vaccines hota hai, but here we are talking about only 2. What are the 2 main categories?
What name is 2 main categories of vaccine?
With 1 example in each case. 1 example in each case. The first category you have to write vaccines obtained from killed germs. That's the first category. Vaccines from killed germs. And next is vaccines from live and weakened germs. Live, they are living, but already treated with chemicals or heat. Live, weakened germs. Write an example. Yes, Levan Bolo. These are done. Ostia and ossiculum.
I didn't get the audio.
Ostia, ossiculum. Ostia are small pores present all over the body of sponges. Ostia are small pores present all over the body of sponges. Small pores present all over the body of sponges, through which the water enters. Through which the water enters. Through which the water enters. Next sentence, ossiculum.
This one about
Okay. This time, Okay, we'll look at the next part. Nitrogen fixation, I'll explain you next class. Nitrogen fixation is a detailed concept. Okay, moving to the next part. Bacteria in industries. Very fast we can go through this. Bacteria in industries, what you see there? What is the role of bacteria in industry? Number one, flavoring of the tea. You know what is flavoring of the tea? The tea leaves. Okay, why certain type of tea leaves we get 1 kg 2,000 rupees and certain type of tea we get maybe 1 kg just 200 rupees also. Why like that? Because what type of bacteria is involved in the processing makes it possible, and that also is responsible for the flavor. So certain type of tea we come up with flavor is not that much, but certain type of tea the flavor is so good, the aroma is so good. So now that is what is called flavoring. So what is flavoring? There are certain type of bacteria involved in. There are certain type of bacteria involved in processing, drying, and in that time the bacterial cultures are being used. Leather tanning. Do you know what is tanning of the leather? Coloring. The leather, basically, if I talk about the hide of the animals, no, the treated hide of the animals. So this hide of the animals is what you are wearing as shoes, wearing as belts. You are taking wallets, bags. So now that we are talking about this, what is a characteristic color? There's a characteristic color, either yellowish or brownish or golden brown. So there's a characteristic color. So this color is called tannin. But actually the word tannin comes from the word tannins. Tannins are a plant product. That is. But this tannins, as it is, can we just brush them on the hide and let it stay? No, this is a particular process of treating a bacterial culture. These hides have to be treated in those tannins in the presence of different types of bacteria. Then only the color becomes fast. Did you ever experience that you're wearing the shoes and the color comes out from the shoes? The belts, no, the bags, no. But yes, when we are talking about the color, it is only for the polish, but not the actual color is not coming out because the color is already fast. The process of making the color or coloring the hide, that is called tannin. Next, if we talk about underline the word botulism. What is botulism? Food spoilage. What is the meaning of food spoilage? Quite a number of times you also want to experience the canned food. The tin food, it comes in some packs, no? The tin ones, or the juices, or any type of those canned food, variety of canned foods nowadays we come across. Now these canned foods, do we consume them after their expiry? Not at all. And it is also advisable that almost a week before their expiry also we should not consume to be safer. Because it is written 25th November. Is it actually that the bacteria will start growing only on the 25th November? No, it will start growing much earlier also. It's always safer for such type of products to stop using them at least 4 or 5 days earlier than this. Then what will happen if by chance we consume also? We do consume also at times. By chances of we go outside. After expiry also, and if not, we may not try to consume, but when you go outside, we never know what it gets out. So that's why. Then we suffer from what? Indigestion, extreme stomach problem, and indigestion and stomach ache. So that is what we are talking about. So underline that word food poisoning and botulism. So this word we will come across. Now the next class we are going to come across how to learn about the preservation. How do we learn about preservation? For this side also, we'll have to continue with some more questions. Okay. Your exam is next week. No? Or this week only?
Yes.
This is Saturday. Next Saturday are you there?
Yes.
Next Saturday. So we'll do some more questions on invertebrates vertebrates. And for you will see, for you will see if we can keep one lecture so that this chapter is very and question solved because I am really not understanding what you are following. Questions we are solving right now. But at least we should discuss also. Then only I'll be able to understand how much we have to follow. Okay. Let's see.
Hey,
जिसका माया वाला करेगा
अनीश को भी
हाँ नहीं कोई आगे ऐसे खाते हैं क्या आएगा
हाँ
मैंने कभी खाया नहीं है ना
अरे
इस बार खाया
अरे ना देख के कैसे होते हैं अरे
हाँ एक नया फैक्ट्री में लाइट
अरे मास्टर मेरे बेटे देखेगा
उधर ही ज्यादा नहीं
Only